Hi, everybody. I'm Hal Foster. I'm the BioNews Services correspondent for FA News. And with me today, I have Dr. Mark Payne. He's a pediatrics professor at the University of Indiana, and he specializes in the cardiac side of Friedreich's ataxia. And we're going to talk today about what he does. He's working right now on a new therapy approach to FA called protein replacement. So Mark, let me just start. I'll ask you about your background a little bit later, but let me just start with the stuff you're doing. So tell me what you're doing these days on protein replacement,
right?
And what you see it, where you see it headed.
Okay. So the concept behind protein replacement therapy really looks at diseases that have a well-identified single gene defect, and Friedreich's ataxia is one of them. So the gene defect is known, and the defect, the disease, is due to lack of a protein, frataxin. And as the patients age, then symptoms appear. So the concept is, can we simply replace the protein that's missing, in this case in mitochondria? The trick is to get the protein from an injection site or from an oral intake or from an IV infusion, for example, to where it needs to go in the body and then where it needs to go inside the cell. And in this case, frataxin is a mitochondrially targeted protein, so it has to get into the cell, into the tissues, and then go into the mitochondria where it is properly processed and does its job. There are multiple ways that you can achieve protein replacement therapy, one of which is to create a replacement protein and engineer it so that it will cross all of these membranes and move into mitochondria, and that's the approach that I take. Other approaches would be to institute viral gene therapy, for example, which is going to produce the protein inside the cell, and then it would hopefully replace the missing protein as well.
And you're talking about mitochondria. That's the energy component of cells, right? Can you explain?
That is correct. So mitochondria actually have a lot of jobs inside the cell. One is that they produce energy for the cell, and there are specific organs inside the body that use a ton of energy, one of which is the eye, the heart, certain aspects of the brain use a lot of energy. And without adequate mitochondrial function, over time, these organs, these specialized tissues will fail, and then disease becomes apparent. In order to mimic these diseases, we use mouse models, or mice that have been genetically engineered to have certain aspects of the disease that we want to study. And I use these models for my protein replacement experiment work.
Okay. When did you start protein replacement work?
About 2000.
Okay. Oh, so
it takes a while. So there was the initial discovery that protein replacement would work using fluorescent proteins, for example. And this works because there's a cell-penetrant peptide that's attached to the protein, moves the protein across the cell membrane. So without it, this doesn't work at all. And we could show that with fluorescent proteins, we could get our protein into the mitochondria of all tissues in the mouse, and that was pretty exciting. This was based on an earlier publication from Steve Dowdy's lab at Washington University, and we followed up on this. And then from that, we began to look at different disease states, and mitochondrial diseases are actually quite common. On an individual basis of specific diseases, they're rare, but as a group, they're quite common. So we could show that in mice that had lost the frataxin gene expression, which we got from Helene Puccio, we could show that if we replaced it, the mice lived much longer and their heart function was much better. So that was very encouraging because we were putting a human protein back into a mouse, which is not the normal protein for the mouse to have, and yet it still would work. Didn't rescue them all the way, but it rescued them enough to show proof of concept, allow us to develop intellectual property behind it, develop grants from the NIH, from the American Heart, from the FARA organization, and move the work forward. And then eventually we were able to achieve a company that has funding through New York and from the NIH to move this forward into commercial production or commercial development.
Is it in clinical trials now?
It is not. It's at the point of called clinical development, where we're working to manufacture the drug, which we've done, and we are working to develop the pre-IND or the FDA application, for example.
Okay.
So there's a lot of administrative steps that are required to actually move towards a phase 1 and phase 2 trial. And we're working very hard to achieve that.
Okay. And you specialize in the cardiac side of Friedreich's. For those people who don't know about it, would you explain why this is a problem, how big a problem it is, and so forth?
So Friedreich's ataxia becomes noticed first because kids and adolescents and teenagers in particular suddenly lose their ability to run and play and walk, and they develop a gait that looks like they're drunk, which is not right. But at the same time, what became very apparent when I started working in this field, and again in 2000, was that yes, they lose their ability to move and control their bodies, but their hearts were very sick, and in about half the cases, the patients die because of their heart disease, and they develop a very thick heart. And in cardiology, we see this. There are many diseases that cause a really thick heart, but in this particular case, the hearts develop a very unusual feature. They get very thick. They continue to beat very strong, but what really happens is they're having trouble filling with blood, so that it makes them hard to get enough blood flow out to their body. It's called preserved ejection fraction with heart failure. Probably not so important. What happens, though, and what really is important, is that, yes, they've got the neurologic findings, but the heart disease is somewhat silent. And so it's very important for any patient who's just been diagnosed to also see a cardiologist, a pediatric cardiologist if they're children. And they'll be very familiar with the disease. We don't have a specific treatment for it. It's going to require either protein replacement therapy or viral gene therapy, or perhaps another therapy that is experimental at this time as well. But we can monitor this, and we can follow the side effects of having such a thick heart and hopefully prevent any symptoms or side effects.
Okay, very good. I'm talking with Mark today on the sidelines of the International Ataxia Research Conference in Pisa, Italy. And we're in the second day of the conference. It's going to be a 4-day conference. Mark, what have you seen so far? There's a lot of new stuff going on here.
Yeah.
What's exciting to you about the conference so far?
Yeah, there's been some really exciting stuff. One of the talks today was on exercise. So this was one of the physiologists at University of Virginia. And he had been working with one of the mouse models, and there are multiple mouse models of Friedreich's now. This one's called the Kiko mouse model. I was so impressed that he could exercise the mice and show that with exercise, they improved their mitochondrial function. They improved their ability to maintain normal muscle strength, and that was very exciting. Obviously, as cardiologists, we all tell our patients, yes, exercise. You'll help avoid some of the complications of Friedreich's ataxia, diabetes being one of them, and you could show the mice use glucose much better. But this really provided some of the basic science evidence that exercise was beneficial in this disease. And with this in mind, that lets us go back and structure a clinical trial looking for very specific endpoints in Friedreich's ataxia that would justify telling our patient, yes, you need hard exercise, you need to keep your heart rate above a certain level, and you need to be tired when you're done exercising. And this may improve their outcome. It won't cure the disease, but it may help them maintain mobility and avoid problems later on in life.
Very good.
And that's very exciting.
Tell me where you just a quick rundown of where you've gone in your research, and just a brief rundown from where you started and how you got here.
That's a long road.
Yes.
So the work really started out. The work started out based on my postdoctoral basic science work, where I was studying how proteins target mitochondria and move into mitochondria. So it was it's completely clinically detached understanding of basic events. But then the work began to evolve into what diseases have problems arising from mitochondria in the heart and missing proteins, and then can you repair that, or can you fix that defect? And it became apparent that it could. So this work has moved from very basic science, and it's built completely on the work of other better scientists than I. And I have to give credit to them, but this work has moved from pure basic science into a field called translational biology. So at this point, we're using mouse models to mimic human disease, and then using our basic science to see if we can improve the mouse model. and then from that extrapolate into the human. So within my own area of research, I've done both basic science investigations as well as clinical in the patient. So one of the other studies that we have recently completed was to look at the heart's ability to burn specific fuels in the patients. And we used an approach called positron emission tomography, which is a clinically useful tool, in which we could calculate how much glucose the heart's burning, how much fatty acid the heart is burning. And then we used our mouse models to validate that by using our mice in a special mouse PET scanner as well. So this is a very difficult area to work in because basic science has a very well-defined mechanism to fund it and to move it forward, and clinical science has a very well-defined mechanism to fund those. But what's really hard is moving investigation from basic science into human beings. That's called translational research. And that's a very challenging field to work in because you have to be able to speak the language of basic science and speak the language of clinical science as well.
Thank you. Enlightening. I'm glad that things are moving forward in protein replacement. And thanks so much for being with us today. I hear great things about you from all the rest of the people here.
I enjoy this meeting so much. There's so much energy that comes out of this, and you come away with new ideas, and it really charges you up to come to these meetings.
Thank you so much.
Thank you, Hal.
Bye.